Okay, here we're going to go through the PowerPoints. Hopefully you watched the previous video explaining the theory behind this. Now we're just going to look at the parts and talk about the chemistry, how to make the chemicals. These would be the things that you would do in the lab if we were meeting. So we had previously talked about PCR, agarose gel is what agarose gels are what we're talking about today. Probably need to go back and give you another video on PCR. But this is the apparatus. If you watch the previous video, this is like the container that we're going to kind of produce our gel and run our gel in. And it's designed this way where you tape up the ends so that you can form the gel and then take the tape off, and the gel will just slide right out, and so you can move the gel around after it's formed. But one of the hardest parts of this is to tape this up and not let it leak. And if we were in lab, we would be spending an hour probably taping up our apparatus and trying to make sure it isn't leaking. There is an art to taping the ends of these little trays so that they actually can form your agarose gels. So we talk a lot about this when we have regular lab. Of course, we don't do that here. Now, to make the little wells, you put what's called a comb in there, and it's kind of like a mold. And then, so you pour the liquid in, put the comb in, then you let it solidify, and then the comb is the blue thing here. And then you just pull out the comb, and you have little holes left where the comb was. All right, it left little holes in there, so they're just like little I don't know. It does kind of look a little bit like a comb you comb your hair with, but the bristles or whatever are wider, and they make little little holes, wells. We call them wells in the gel. And after it solidifies, you pull this out. It's like a mold. Pull it out, and voila, you have little wells in which you can put your DNA. Now we add a dye to the DNA, something that sticks to the DNA and will cause it to fluoresce. And so this is the name of it, ethidium bromide, and we add it to the DNA to make sure that the DNA can be located, and I'll show you what that looks like. This is showing you how some of the chemistry of that. So you need to memorize this stuff. There'll be written questions about this. This is the solutions we would have made in class. We need 50, that's micrograms of per milliliter of this, and etc. Just know these. Just literally memorize this. Let's see. You'll need to know this. This is just the ratios and things like that for making sure that the chemical processes work correctly in our gel. So you'd have to know all of this ahead of time before you went into the lab and actually did the experiment. And in normal class, we would be going over this in class and then actually doing all of this. Now, electrophoresis is what I talked about in the previous video, and you need to understand everything I said in that one, because there will be questions like that, and hopefully the video I gave you, especially like positive and negative attraction, repulsion, that sort of thing, will help you out. It uses an electric field, of course, and here's how you would mix everything up. We used the ethidium bromide as a dye, but we're going to show here in a second. We'll use a buffer as our electrolyte. So make sure you read through these PowerPoints. They'll be on the OneDrive. And here's your buffer. It's TAE. It's tris acetate EDTA, or TAE for short. So you will need to add that's the running buffer. The loading buffer it helps you. It doesn't help that much, but you have to do it. You have to be able to. It's to help you see. It makes it blue so you can see it. That's about all we can say. At least see it before it's run. I'll show you. There'll be some pictures here. So here are all the chemicals you would have added and things you would have done beforehand. So make sure you know these for the test. Here's our little apparatus, and then here we're hooking it up to the power supply, the electricity. And notice that up here, your DNA is on the same side as the negative charge, so that means these are going to repel. DNA is negatively charged. This is positive, so it's going to be attracted down here. So it's going to pull the DNA through the gel. So if you load it here, it's going to pull it in this direction. If you let it keep going, it'll just run off the end. And here's your electrolyte, the purple here. This is the liquid, and you pour just enough liquid to cover the gel, not too much more, and that's all you need to do. So you put your gel in here, you fill this with liquid so that it just covers the gel. Then you turn on the power, and then you just wait for it to pull the DNA through the gel. All right, and here somebody with a pipetter, and the mixture, the DNA with all the other chemicals and everything, that's the stuff in blue, and you just put it in these little wells. All right, as you can see, this one is empty here at the end, and then they put the pipette in and pipette it in. Here it's getting filled, and here it's completely filled. And we usually take time to practice this a little in class when normal time. And then here, it's after the DNA is run, you put it under UV light, and the dye we added essentially fluoresces, and this protein binds to the is it protein? It isn't. The chemical binds to the DNA. It intercalates is the term, and binds to the DNA and fluoresces when you put it under UV light. And so you can very easily see where the bands are, and this is where your DNA is. So this is probably a ladder here. This is telling us, you probably would have known that, oh, this is 50, 100, 150, 200. And this would be telling you that, oh, all three of these are, you know, maybe 100 or a little bit more than 100. This one is closer to 100 here. And these differences are telling you that these are not from the same samples. The DNA was different. It cut in different places. So I suspect that this is a ladder, and then this is a ladder as well, and because this ends about where this one starts, so therefore these are two different ladders. And you know, so this one is maybe 50, 100, 150, 200, and maybe here you start at 250, then very faintly, you know, 300, and then maybe this is like 400 and 500 up here or something. So this is telling you the sizes of these, but it's definitely showing you that these samples, if these were from different organisms or different people, or maybe different mutants. So maybe this is wild type, and then here are two other mutants. Here a piece is completely missing out of this one. Here they've been cut slightly different, something like that. So just keep in mind that this is what you should expect. Now this one that's thicker, that's telling you there's more DNA there. These are probably all about the same quantity, so they all show up as equal size and brightness. But when you get something like that, maybe you cut it and the two pieces were equal in size, and they might show up as a bigger blob, a bigger band that's thicker, and it also probably would be a lot brighter. So we move on. Here is essentially your protocol if we were doing this in the lab, and this is probably testable. Here are the little tubes that you would have mixed everything in, and of course we would have, in the lab we keep different colors to make sure you keep everything separate. That's why we've got pink and blue here, and here's what you would have put into these. And here's how you would have arranged it. It was arranged really nicely, so it tells you exactly what you put into each vial, or each tube. So you have 8 tubes, and here's what you put in, and then we're going to run all of these in our in the gel, and then we're going to compare what we get. So this would have been 2 wild types. You know, so you usually run multiple samples, and we have controls and all kinds of interesting things here. And this is the chemicals we would have put in here, the amount of DNA, etc. And here, of course, this is your ladder. All right, giving the example. So here's 250. So it's telling us that in this sample we have one band and it's just a little less than 750, probably maybe 700 somewhere in there. All right, and that is it.